So dear students, after the successful completion of all the chapters in second PUC in all chapters na mugiskottidini 16 chapters na mugiskottidini. First PUC alli 7 chapters igagale explain madidini. The 8th chapter adanna madbekagide, the cell, the unit of life anta. Cell, the unit of life anta. So first cell described by Anton van Leeuwenhoek described the cell, whereas the nucleus is described by the Robert Brown. In 1838, Malthus Sleeden observed the cells in the plants which constitute tissues, and then Theodore Schwann, a Briton zoologist in 1839, not only observed the cell membrane in animal cells, also observed cell wall in the plant cells by the Briton zoologist. Whereas the Malthus Sleeden, German botanist. So Malthus Sleeden, German botanist, and the Theodore Schwann, the Briton zoologist, proposed the cell theory. According to the cell theory, all the organisms are made up of structural and functional unit of life called cells. But that cell theory modified by the Rudolf Virchow in 1855, he proposed omnis cellula cellunae. That means all the cells are originated from the pre-existing cells. Now the modified cell theory is all the living organisms are made up of structural and functional units called cells, and these cells are originated from the pre-existing cells. And then the cytoplasm, it forms the arena of all the cellular activities. Within the cytoplasm, different cell organelles are present. So like the endoplasmic reticulum, mitochondria, Golgi complex, ribosomes, in animal cell, centrosome, centriole, etc. The difference between the plant cell and animal cell, you know very well. The plant cell has cell wall, no cell wall, and here plastids present, no plastids. In animal cell, here no centrosome, that is centrosome. No centrosome, and centrosomes are present in animal cells. These are the major differences, and large size vacuole, small size vacuole. These are the major differences between the plant cell and animal cell. In general, the cell, the smallest one being the viruses. Now it is not a cell, but the viruses are small in size, 0.02 to 0.2 millimicrons mu in size the viruses. And coming to the mycoplasma, 0.3 millimicrons in size, and the PPLO 0.1 mu in size. The bacterial cell 1 to 2 millimicrons in size, whereas the eukaryotic cell is 10 to 20 millimicrons in size. The largest cell is the ostrich egg cell, and the smallest cell in the human beings, RBC, that is 7 millimicrons in size. So these questions also will be asked usually. So first in the textbook they explained about the bacterial cell. The bacterial cell consists of cell wall made up of peptidoglycan, or it is also N-acetyl galactosamine. So that is about the cell wall. But the cell wall in bacteria consists of a slimy layer called glycocalyx, which is thin, and the thick layer called the capsule. And in the cytoplasm of the bacterial cell, here only the incipient nucleus or nuclei present. Cell organelle is like endoplasmic reticulum, mitochondria, Golgi complex are observed. Only ribosomes are present here and there, the ribosomes with the messenger they forms a polysome. As we know very well, the ribosomes are discovered by the Faraday in 1953, and ribosomes are chemically nucleoproteins. They are unbounded. These this is very important. No membrane for the ribosomes. Ribosomes are of two types, 70S type and 80S type. 70S type subunits are 30 and 50. S stands for Swedberg unit, and 80S type subunits are 60 and 40. Values are not additive. And then the 70S type present in the prokaryotic organisms, 80S type present in the eukaryotic organisms, and ribosomes with messenger RNA forms a polysome or polyribosome. This is also very important and play an important role in photosynthesis. Only 70S type of ribosomes can be seen in case of the bacteria. That is very important. And those bacteria which can be stained by the Gram stain called Gram positive bacteria cannot be stained by the Gram stain, that is Gram negative bacteria. So in the absence of mitochondria, mesosomes play an important role and help in respiration. These are the extensions of the plasma membrane present in the bacterial cell, and this consists of the vesicles, tubules and lamellae. Vesicles, tubules and lamellae, the mesosomes consist of all these parts. It helps in the cell wall formation, DNA replication, respiration, and then secretion, increase of enzymal content and increase of the plasma membrane for the surface area. These are the various functions of the mesosomes, and that is about the bacterial cell. And in addition to bacterial cell, in addition to these parts, that is the flagella present. This flagella made up of a protein called flagellin, and they have the filamento-hucco-basal body. Filamento-hucco-basal bodies are present, and in some bacteria there are fimbriae which help in the anchoring to the host or even to the rock. Some confusion may be there, and fimbriae also present in the finger-like projections, that is in female reproductive system also. And there is pili made up of proteins, and pili is said to help in the DNA transfer of DNA, that is during the sexual reproduction in the bacteria. And apart from all these things, the bacterial cell, few bacterial cells, they have inclusion bodies like the phosphate, glycogen, synophysian granules, and gas vacuoles are present in green purple bacteria, even in blue green algae. So this is about the in detail the bacterial cell. Coming to the cells, eukaryotic cells. In eukaryotic cells, in algae, this algae made up of the plant cells in algae composed of galactons, mannans, cellulose. This is also few algae consists of galactons, mannans and cellulose. The primary one composed of chiefly the cellulose. The middle lamellae consists of calcium pectate, and the secondary one also composed of cellulose. And here the middle lamellae is in connection with the other cell walls through plasmadesmata. This is very important. So far not asked. So this is about the cell wall. Cell wall offers protection. Next to cell wall is the plasma membrane. Plasma membrane first noticed in 1950 in the RBC. And this plasma membrane, it consists of the two protein layers and the lipid layers. The protein layers, lipid layers are sandwiched between the two protein layers, and then there are proteins, membrane proteins and integrated proteins. This is also very important. So far not asked. The lipid layers they have the polar ends and the non-polar ends. Non-polar ends composed of polysaturated hydrocarbons, and they are all chiefly made up of the lipids which are present are the phosphoglycerides. So the cell membrane in total consists of about 52% of proteins and 40% of lipids. This is also very important. And the fluid mosaic model that was proposed by Singer and Nicholson in 1972. This is very important. 1972, you know very well. Stanley Cohen and Bayer also first recombinant DNA they produced in 1972, and similarly in 1972 Singer and Nicholson proposed the model of the fluid mosaic model of the plasma membrane, and 1972 is also known for the Wildlife Act that is implemented in the year 1972. And then Singer and Nicholson proposed the model of the fluid mosaic model of the plasma membrane. According to this model, protein molecules either partially or wholly embedded within the lipid layers, giving an appearance of mosaic. Protein molecules either partially or wholly embedded within the lipid layers, giving an appearance of mosaic. This is very important. And then there are polar ends and non-polar ends. Lipid layers are present throughout, but the protein molecules are embedded within the lipid molecules, either partially or wholly, giving an appearance of mosaic. The interior region is fluid in nature. That is very important. This kind of fluid nature helps in the cell growth and intercellular movements and also secretion, endocytosis. So far not asked. Within the cell membrane, divisions takes place, increase in the chromosomes takes place. That is endocytosis. It helps in cell division. That is the important, because the fluid nature does it helpful for the cells. As you know very well, the plasma membrane offers protection, regulates water composition, plays an important role in osmosis, and also phagocytosis and pinocytosis, and also the transport across the membranes, facilitate transport. As we know, this is also very important. Polar molecules, that is the water is a polar, cannot pass through non-polar lipids. They require certain proteins to carry this, that is active transport at the expense of, that takes place at the expense of energy, and it is also a facilitated transport. And in the next chapter we will study what are all the different types of facilitated transport, diffusion or the active transport. Anyhow, passive transport takes place without the expenditure of energy. Active transport requires the energy. So that is about the cell wall and all those things, cell wall and cell membrane in eukaryotic cells. And coming to the in cell, after the plasma membrane in animal cells as well as in both plant cells. So the other cell organelles present are endoplasmic reticulum, Golgi complex, and then the vacuoles, lysosomes, mitochondria, chloroplast, ribosomes, as I explained earlier, ribosomes, and centrosome, about the cilia and flagella. So all these things are the cell organelles. So there are two systems, that is endomembrane system, where the functions are coordinated. So endomembrane system, endomembrane structures are the number one, endoplasmic reticulum, and then Golgi complex, lysosomes and vacuole. These are the four wherein their functions are coordinated. Whereas in case of mitochondria, chloroplast and peroxisomes, their functions are not coordinated. They do not come under endomembrane system. This is also very important. So coming to the endoplasmic reticulum, it is the net-like structure which is present in the cell. So much portion of the cell occupied by endoplasmic reticulum. The innermost layer of the plasma membrane, outermost layer of the nucleus extended as endoplasmic reticulum. And endoplasmic reticulum also consists of two major parts, that is the luminal part and the extraluminal part, that is the inner layers, the luminal inside, and extraluminal, that is the cytoplasm, so far not asked. And the endoplasmic reticulum in touch with almost all the parts, like especially with the Golgi complex, the cis associated with the cis part of the Golgi complex. And there are two types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum will have ribosomes which help in protein synthesis, whereas the smooth endoplasmic reticulum doesn't contain the ribosomes, but hence it play an important role in the synthesis of the glycolipids, lipids especially, as well as the steroid lipids also synthesized by the smooth endoplasmic reticulum. And the endoplasmic reticulum is in contact with the cis portion of the Golgi complex. Golgi complex 0.1 millimicrons to 1 millimicrons in size and consists of plate-like structures called cisternae. Cisternae size is 0.5 to 1 millimicron, not the Golgi complex one. So here the Golgi complex helps in the packaging the materials. It packages the materials. Endoplasmic reticulum, as we know, the rough endoplasmic reticulum have the ribosomes. They synthesize the proteins. These proteins are carried to the cis portion of the Golgi complex, wherein the formation of the glycoproteins and glycolipids takes place. That is very important. The formation takes place in the cis region of the cisternae of the Golgi complex. Then they are transported towards the trans region, and from the trans region to different parts where they require. Thus, this is about the Golgi complex. And coming to the lysosomes, these are the single membraned structures, and they are meant for intracellular digestion. They digest carbohydrates, proteins, lipids. Not only these things, but also nucleic acids. That is very important. But they are active at lysosomes secrete certain enzymes which are very active at acidic pH. That is also important. Next to this, vacuoles. Vacuoles occupy about the 90% of the cell portion, the vacuoles, and then the vacuoles, you know very well, the vacuole layer, vacuole composed of tonoplast. So sometimes the tonoplast, against concentration gradient, the vacuoles, they absorb certain materials. And in amoeba, etc., full vacuole and contractile vacuole, which maintains osmoregulation, can also be seen. And 90% of the plant cell, there are only vacuoles. Large size vacuoles present in plant cell and small size vacuoles present in the animal cell. Coming to mitochondria, which is very, very important. Mitochondria are called powerhouses of the cell, as we know very well. Mitochondria are composed of two layers, inner layer and outer layer. Inner layer is inwardly projected to called cristae, whereas the space between these two is called perimitochondrial space. It's also called outer matrix. Outer matrix, very important. And then outer matrix. On the walls of the cisternae, that is cristae, there are F1 particles or elementary particles or Racker's particles are present. In the inner matrix, single circular DNA, 7 ribosome, RNA, few molecules present, that is important, not given so far. Few RNA molecules, single stranded DNA, and then the 7 ribosomes. Certain enzymes are present in the inner matrix of mitochondria. As we know very well, they are called, why they are called energy I mean the powerhouses of the cell means the respiration. There are two types of respiration: aerobic and anaerobic respiration. Aerobic respiration, aerobic respiration involves the glycolysis, Krebs cycle, and terminal oxidation. Enzymes required for Krebs cycle are present in the inner matrix of mitochondria. This results in the formation of NADH2 and FADH2, as we know very well. And then this the energy released during glycolysis and Krebs cycle especially stored in the form of NADH2 and FADH2. Transfer of electrons takes place from these compounds along the chain of cytochromes. I will explain this in the respiration in detail. And then during the transfer, energy is released. That energy is used for the formation of the ATP that takes place in the presence of molecular oxygen. Hence it is called electron transport system and electron transport chain, terminal oxidation, biological oxidation. Finally oxidation takes place, resulting in the formation of water. So these things occur in the inner membrane, that is the cristae from F1 particles, F0 forms the channel for these things. And the H plus ions during the respiration will be released into perimitochondrial space, which is very important for your NEET examination. Whereas during the photosynthesis, H plus ions are released into lumen of thylakoids, whereas the H plus ions during the respiration, oxidative phosphorylation, that is released into the space between the two layers of mitochondria, very important for your NEET examination. Coming to the chloroplast. Chloroplast, just like mitochondria, composed of two layers. Inner layer and outer layer. Chloroplast cells have green colored grana and colorless stroma. And the grana composed of plate-like structures called thylakoids, and these grana interconnected by stroma lamellae, and there will be stroma. And these are the sites for the photosynthesis. And this chloroplast, single chloroplast, can be seen in Chlamydomonas, whereas the number of chloroplasts is 20 to 40 in case of the mesophyll. And we know very well the photosynthesis takes place in two steps: light reaction and dark reaction. Light reaction takes place in two steps, PS1 and PS2. PS1 takes place in the grana thylakoids and also stroma lamellae, whereas the PS2 takes place only in grana thylakoids. This is also very important, resulting in the production of the ATP and NADPH2, and these end products are utilized during the dark reaction, which takes place in stroma of the chloroplast, resulting in the production of the glucose. That is very important. And here in the stroma, 70 S-type ribosome, but double-stranded DNA present, along with certain enzymes required for the dark reaction, which is very important. And agranule chloroplasts are present in the bundle sheath mesophyll of the C4 plants, also very important. And the plastids. Before explaining actually chloroplast, you should have been explained the plastids. So plastids are the colored pigments which are present only in the plant cell but not in the animal cell. These plastids are of two types: colored plastids called chromoplastids, and then green colored plastid called chloroplast, and colorless plastid called leucoplast. Leucoplast, which store starch or carbohydrate, that is called amyloplast, and which store proteins called alliloplast, and which store oil called helioplast. This is also very important for our examination. And coming to the flagella, this flagella, it consists of central 9+2 arrangement, two central microtubules. They are connected by the bridge, and around this there is a central sheath, and there are peripheral doublet tubules are present, microtubules are present, and then doublets, peripheral doublet are also microtubules can also be seen. These are the various parts associated with the flagella or the cilia. They show, and the core of this cilia flagella is also called axone. Whereas the centrioles present only in the animal cell, which consists of the central hub-like structure and spike, the spoke-like structure. And these they have the basal bodies of cilia, flagella, basal bodies of spindle fibers. Spindle fibers, as you know very well, they form the spindle apparatus, and gets attached to the centromium. So this is about the different parts of the various cell organelles. Whereas the other important cell organelle is nucleus. Nucleus, it was described by Robert Brown in 1831, which is called controlling center of the cell. Nucleus consists of two layers, the inner layer and outer layer. Outer layer extended as endoplasmic reticulum. The inner layer. Space between these two is called perinuclear space. And within the karyolimb, within the nucleoplasm, nucleolus present. Nucleolus, it is the site for the production of ribosomal RNA, which is very important so far. And there will be chromatin fibers. So chromatin term given by the Fleming. And then the chromosomes are the colored bodies which are present in the karyolimb. Prior to this, there is chromatin, and chromosomes consist of arms which are held at a point called primary constriction, which is an unstrainable portion. Surrounding this, there is kinetochore, and then the spindle fibers produced usually gets attached to this kinosomes or kinetochore usually, and this chromatin, and then there are certain chromosomes in which the dicentric chromosomes. In addition to primary constriction, secondary constriction is present. Portion distal to the secondary constriction called satellite cyanoacid thymonucleonico. Such chromosomes are called the satellite chromosomes. Based on the position of the centromere, chromosomes are classified into metacentric, where the centromere present at the center. This is also very important. Two equal arms. Submetacentric, just away from the center. There will be a long arm and a short arm in submetacentric. And in telocentric, only one arm present, a long arm that is present at the tip. Just away from the tip, called acrocentric chromosomes. Just very long arm and a very short arm can be seen. So this is about, based on the position, the chromosomes are classified. Based on the number of centromeres, monocentric, dicentric and polycentric chromosomes. So thus they are classified. The last aspect here are the microbodies. Microbodies contain enzymes, and they are present in both plant cell as well as in animal cell. So this is about in detail about the cell, the unit of life. Please read sentence by sentence. That is very, very important for your NEET examination. Thank you.